I would like to thank also Dr. Fayhan for his great deep and autoimmunity mechanisms. Now we will go to something more simplified, talking about basic immune laboratories and what usually we do there, especially for rheumatologists. So the allergist, and usually immunology labs provide an extensive range of laboratory investigations and clinical support to assess the diagnosis, surveillance, and treatment of infectious, non-infectious immune diseases, allergy, autoimmunity, and also they have an extreme help in solid and organ and bone marrow transplantations. So what are the main methods used in immunology labs? We have what we call ELISA, enzyme-linked immunosorbent assays. We have the new form of immunoassays we call chemiluminescent microplate or microparticles immunoassay. We have nephrometer. We have Western blot, or what we call them immunoblotting. We have also immunofluorescence microscopy, flow cytometry, immunoperspation, and immunoelectrophoresis. As everyone knows, ELISA are extremely used since a while in all laboratory testing, especially in immunology serology aspects. And basically, they have different types of immunoassay. We have direct immunoassay, which we directly detect antigen or antibody. We have an indirect immunoassay, ELISA, which also we have a type called the sandwich ELISA, which use two different types of antibodies, and we also have a competitive ELISA. Another type of immunoassay, we have the ChEMIA, or what we call the chemiluminescent microparticle immunoassays. These assays utilizes the isolation of antibody-antigen complex on a solid phase surface of a small beads called microparticles. Basically, the ChEMIA is considered to be an advanced and modified form of an ELISA technique. They are considered to be more sensitive than ELISA, but the issue with ChEMIA that it might give you a false positive due to its extreme sensitivity compared to ELISA. Another method that is used frequently in immunology labs, nephrometer, which is a technique used in immunology labs to determine the level of several blood plasma proteins. Examples are like the complement 3, C3 and C4, and they are usually low in active lupus. We have also the CRP and antibody concentration, the immunoglobulins, IgG and IgM. The major histocompatibility complex, MHC, or in human we call them HLA, the human leukocyte antigen. They are located in a short arm of chromosome 6 in the humans, and these HLA encodes MHC proteins that are essential for antigen presentations. HLA typing mainly can be used for tissue typing, for organ and bone marrow transplantation, and they are also used for disease association, such as the HLA B27 for the AS, which can be found in around 90% of these patients. It can be used also to diagnose or help and diagnose celiac disease, to either DQ2 or DQ8, and also in Behçet disease, which are HLA B51. HLA typing can be done using several methods. We have the most frequent one, which is called the SSO, or the sequence-based specific oligonucleotides. Features of this type of technique, it's a low resolution, so only 2 digits can be identified. All patients and matched related donors are typed for HLA A, B, C, which are class 1, and also HLA class 2, which are DRB1, DQB, and in some certain area DP also, using the SSO. It's considered to be a Luminex method. Another type of HLA typing is the SSP, or what we call signal-specific primers. They are also considered to be low to medium resolution, so we can have up to 4 digits of HLA alleles. Usually they are used to resolve any ambiguous result arising from other methodologies, especially in the SSO. Other techniques which can be used for HLA typing is the SPT, what we call sequence-based typing. They have a higher resolution typing, which can give you up to 6 digits compared to SSO. It's a sequencing-based method, so this is used to determine HLA high-resolution type, especially for stem cell transplant patient and their selected unrelated donors usually. Sequencing is also used to resolve any novel alleles detected by other methods. And recently NGS, or what we call next generation sequencing typing, is been heavily used now in clinical labs, and it has a major advantage that it's a very high resolution, so it can give you up to 8 digits, although in clinical aspects up to 4 is enough. We also have other techniques that can be used, especially for transplantation, is the STR, short tandem repeat, and usually it's a genotype analysis, which is used to monitor bone marrow engraftment after any allogenic transplantation, and we usually call it chimerism. One or more informative STR loci are used to distinguish recipient from the donors. And also in some labs we have the HLA antibody screening, which is also a Luminex-based technique, and it's used to establish the presence of specific HLA class 1 or class 2 reactive antibodies in both pre and post-transplant patients. One of the most frequent immunoassays that used in immunology lab, and it's especially for rheumatologists, is the indirect immunofluorescent for autoimmunity. So basically fluorescent techniques can be used as either direct or indirect. For the direct, it's we are detecting an antigen directly, and by adding a fluorescent labeled antibody to a patient tissue, or wash them, examine under microscopy, and find if there is anything. And usually these type of techniques are used to detect any microorganisms such as viruses in microbiology labs. The other method is the indirect immunofluorescence, and usually it's done by adding a patient's serum to a tissue containing known antigen, wash them, and then add the label antibody. After that, we'll wash again and examine under a fluorescent microscope. Examples of this type is the anti-nuclear antibody, anti-smooth muscle antibodies, anti-mitochondria, and many other more. HIP2 cells for ANA screening is widely used, especially for rheumatology, and this type is still considered to be a very important tool to diagnose the presence of autoantibodies. Usually the ANA screening is done using ranging from 140 to up to 160 as a first dilution of patient. Usually the results are reported as a pattern with an endpoint titer. But we need to put in consideration that up to 10 to 15% of healthy population will stain positive at 1/160. Most positive samples will display a single pattern, but mixed pattern may be observed. Although it's an anti-nuclear antibody, but cytoplasmic staining may also be noted, and it is also reported as a pattern and endpoint titer as well. The important thing that we need to be in consideration that ANA is not specific for connective tissue disease. So here are some examples that ANA might be positive, such as in drug-induced lupus, autoimmune liver disease, chronic liver disease, lymphoma, chronic infection, and malignancy. I will have a couple of slides just to show you how does these patterns look at under the microscope. So here's an example of a HIP2 cell completely negative, no fluorescent staining in the nucleus or the cytoplasm. So this is considered as ANA negative. Here is an example of an ANA homogeneous, which we can see that in the metaphase there is a clear homogeneous staining of the nucleus. And usually the antigens that are related to this type of pattern are histone, double-strand, and single-strand DNA. The association that can be linked to this type of pattern are SLE or drug-induced lupus and also rheumatoid arthritis. But as we said, it is an association, so not all these type of diseases will be linked to these type of patterns. One of the speckled pattern is also one of the frequent seen patterns in autoimmune aspects. Antigens related are as listed in here, SSA, SSB, anti-Smith, RMP, PCNA, and also CL70. The association also, as I mentioned in the previous one, is SLE, mixed connective tissue disease, Sjogren, and scleroderma. Also, we have the nucleolar, although less frequent than the homogeneous and speckled, but are seen a lot. The antigens associated with these type of patterns are the RNA polymerase 1, RMP, and also the PMSCL7 overlapping. The association usually are with scleroderma. It can be seen a lot with scleroderma, and also the overlap of polymyositis and scleroderma. Centromere antibodies also can be seen under the microscope, and the antigens related are centromere either A or B or C, and the association are strongly associated with scleroderma and CREST syndrome. We also have the RIM pattern. It's a homogeneous, but it's more to have a very clear RIM pattern. It's very strongly associated with SLE, and the antigens are usually related with high double-strand DNA level in the patient's serum. As we mentioned before, that ANA is usually screened by immunofluorescence, but there are also lots of other methods that can be done to detect ANA. One of them is the immunoassays, either ELISA or Q-Immunensis. They might have more benefits than using the immunofluorescent. One of them is it's more standardized. It's automated and less variable than HIP2 assays, utilize more or a series of nuclear antigens, as what we can mention them as in a screen, can have sensitivity equal to HIP2 assays, depending on the manufacturer. Maybe less specific, since no pattern can be reported. And some labs use ANA screening by ELISA Q-Immunensis to screen and confirm positive with HIP2 cells and inner profiles, depending on which protocol they are using. We also have some circulating autoantibodies, the AMA, asthma, LKM, and gastric parietal cell. Each one has its unique disease association. The AMA is strongly associated with primary biliary cholangitis, asthma, can be found in up to 70% of autoimmune hepatitis type 1. The liver kidney microsomal can be seen in chronic hepatitis 2, autoimmune. And gastric parietal cell autoantibodies are almost found in all patients with pernicious anemia. Okay, so this is just what anti-smooth muscle look at under the fluorescence microscope. This is the anti-mitochondrial antibody and LKM. And this is the gastric parietal cell. So rheumatoid arthritis testing, I don't want to go in deep details, but we just want to concentrate that we can use either the rheumatoid factor or anti-CCP. Rheumatoid factor are only found in 60% of these patients.